Stems. Plant stems provide a support structure, a support system for the plant body, a transport system that carries nutrients, and a defense system that protects the plant against predators and disease. Stems contain dermal, vascular, and ground tissue. Above ground stems have several important functions. Stems produce leaves, branches, and flowers. Stems hold leaves up to the sun, and stems transport substances throughout the plant. Stems make up an essential part of the water and mineral transport system of the plant. Xylem and phloem form continuous tubes from the roots through the stems to the leaves. These vascular tissues allow water, nutrients, and other compounds to be carried throughout the plant. In many plants, stems also function as storage and aid in the process of photosynthesis. For example, desert cacti have thick green stems that carry out photosynthesis and also store water. Stems are surrounded by a layer of epidermal cells that have thick cell walls and waxy protective coating. These cross sections through a monocot and a dicot stem show the epidermis, vascular tissue, and ground tissue. Vascular bundles, little tiny ones here in the monocot. They're quite large in the dicot. Epidermis, the outside layer. Ground tissue, the gray portion. And the cortex, pith, it's also in dicots. Growing stems contain distinct nodes where leaves are attached, as shown in the figure. Small buds are found where leaves attach to the nodes. Buds contain apical meristems that can produce new stems and leaves. In larger plants, stems have woody tissue that helps support leaves and flowers. How do primary growth and secondary growth occur in stems? Primary growth in stems is the result of elongation of cells produced in the apical meristem. It takes place in all seed plants. In conifers and dicots, secondary growth takes place in meristems called the vascular cambium and cork cambium. Unlike animals, the growth of most plants is not precisely determined, but plant growth is still carefully controlled and regulated. Depending upon the species, plant growth follows general patterns that produce characteristic size and shape of the adult plant. A plant's apical meristems at the roots and shoots produce new cells and increase its length. This growth occurring at the ends of the plants is called primary growth. It takes place in all seed plants. The figure below shows the increase in the plant due to a primary growth over several years. Year 1, this is first portion. Year 2, the primary growth starts from there and continues. In the third year, starts from the end of the second year. This is primary growth for that year. As a plant grows larger, the older parts of its stems have more mass to support and more fluid to move through the vascular tissues. As a result, stems increase in thickness, which is known as secondary growth. The figure below illustrates the pattern of secondary growth in the dicot system. Here's that primary. The secondary growth is the extra thick stem, additionally thick, continue to be thicker to support the increased primary growth. Notice the appearance of bark on these portions. Secondary growth is very common among dicots and non-flowering seed plants such as pines, but is rare in monocots. This limits the girth of most monocots. Unlike monocots, most dicots have meristems within their stems and roots that can produce true secondary growth. This enables them to grow to great heights because the increase in width supports the extra weight. In conifers and dicots, secondary growth takes place in meristems called the vascular cambium and cork cambium. The vascular cambium produces vascular tissues and increases the thickness of the stem over time. The cork cambium produces the outer covering of the stems. Year 1, Year 2, and Year 3. We'll talk about these three in the next two slides. Once secondary growth begins, the vascular cambium appears as a thin cylindrical layer of cells between the xylem and the phloem of each vascular bundle. Right here, the vascular cambium forms between the xylem and the phloem in the vascular bundles. The pith is on the inside. That's in year 1. The cortex is out here. Year 2 looks different. The divisions in the vascular cambium gives rise to the new layers of xylem and phloem. Each year, the cambium continues to produce new layers of vascular tissue, causing the stem to become thicker. This is the secondary phloem getting thicker. And on the outside, the cork cambium has created cork on the outside, bark. And secondary xylem is also forming on the inside. The vascular cambium divides, producing secondary xylem cells toward the center of the stem and secondary phloem cells toward the outside. And the mature stem develops. This inside portion of the xylem becomes wood, and the outside portion, this outside cork, becomes bark in year 3. And this continues, makes the wood wider and the bark thicker. Most of what we call wood is actually layers of secondary xylem produced by the vascular cambium. As wood stems grow thicker, the older xylem near the center of the stem no longer conducts water and becomes heartwood. Heartwood usually darkens with age because it accumulates colored deposits. Xylem in the heartwood, old xylem that no longer conducts fluid but helps support the tree. And then xylem, the sapwood, it contains active xylem that transports water and dissolved nutrients. And on the outside, the vascular cambium, and then we have the phloem, and that includes all of the bark all together, includes the cork cambium and the cork. So the vascular cambium, a meristem that produces new xylem and phloem, increasing stem width. The phloem, a vascular tissue that transports sugar made by photosynthesis. And cork cambium, a meristem that produces the protective layer of cork. And then the cork contains non-functioning phloem. So heartwood, the darker, is surrounded by sapwood, the lighter wood, which is active in fluid transport and is therefore actually lighter in color. This was active in fluid transport. So the xylem sapwood contains active xylem that transports water and dissolved nutrients. When growth begins in the spring, vascular cambium begins to grow rapidly, producing large, light-colored xylem cells, resulting in light-colored layer of early wood. As the growing season continues, the cells grow less and have thicker cell walls, forming a layer of dark, late wood. This alternation between dark and light wood produces what we commonly call tree rings. Usually a ring corresponds to a year of growth. By counting the rings in a cross-section of a tree, you can estimate the tree's age. In a mature stem, all of the tissues found outside the vascular cambium make up the bark, as shown in the figure. These tissues include phloem and the cork cambium and cork. These three, cork cambium, and phloem. As a tree expands its width, the oldest tissues may split and fragment. The cork cambium surrounds the cortex and produces a thick protective layer of waterproof cork that prevents the loss of water from the stem. As the stem increases in size, outer layers of the dead cork often crack and flake off the tree.